All right, hello everyone. So I'm going to talk about some new applications of new B-cell repertoire phylogenetic methods we've developed. But first, why? So the biological basis for a lot of these techniques is the fact that during affinity maturation, B cells undergo mutation and selection to increase binding affinity. This creates B-cell lineages which descend from a common naive ancestor, and this fact allows us to use phylogenetic techniques to retrace this ancestry, to reconstruct intermediate sequences, and to study the types of mutations that occur in B-cell lineages. So the heart of likelihood-based phylogenetic analysis is phylogenetic substitution models. These are models that describe the rates of substitution between different codons or different nucleotides. And the way they work is, given a multiple sequence alignment and a substitution model, we can use an estimation algorithm like maximum likelihood to find a tree topology and set of substitution parameters that describe our data. One of the most interesting is DNDS, or omega, which is the ratio of replacement to silent mutation rates. If this is equal to 1, it's often interpreted as neutral evolution. If it's greater than 1, that's generally interpreted as positive selection, and less than 1, negative selection. When it comes to phylogenetic parameter analysis, the choice of model matters. So SHM is known to be biased by certain hot and cold spot motifs, and this violates important assumptions in most phylogenetic substitution models. And if we don't account for this, it can lead to biases in certain parameter estimates. Which is why we've developed a set of phylogenetic substitution models that incorporate these biases or that incorporate somatic hypermutation biases to reduce these errors in parameter estimation. So if we do simulations of SHM using hot and cold spot bias mutations and try to re-estimate DNDS using the GY94 model, which is a classic codon substitution model that doesn't incorporate SHM biases, we typically overestimate the effect of positive selection. Whereas if we use an HLP type model, we tend to have a much more accurate estimate of DNDS. Another issue is the fact that phylogenetic substitution models are usually made to describe single lineages, whereas B-cell repertoires are made up of hundreds or thousands of lineages. So under a standard approach, we would take each of these multiple sequence alignments and estimate substitution parameters for each of them individually to get our individual lineage trees. Alternatively, what we do is to take all of our multiple sequence alignments for all of the lineages within a repertoire and estimate parameters of a shared model that describes evolution of the entire repertoire, and from that get our phylogenetic trees. So this gives us the ability to estimate substitution model parameters that characterize entire B-cell repertoires. This has several advantages. One of the clearest is it increases our precision in estimating these parameters. So if we look at WRC hotspot mutability estimates for individual lineages, which are shown in these black dots, within a repertoire we can see that there's quite a lot of variation, especially for smaller lineages. And if we look at our repertoire-wide estimate as this blue line, we can see that it represents kind of a weighted average of the types of parameters that we see in the repertoire. If we use simulations to try to assess the accuracy of these parameter estimates, we can see that estimating these parameters for small lineages is incredibly error-prone, almost meaningless. With more sequences, we tend to get better estimates, and with repertoire-wide models, we have far better precision in estimating these parameters. So what can we do with this? One application we found is to take a data set of 27 healthy individuals of varying age and sex and estimate phylogenetic substitution model parameters for each of them. So for this figure, each row represents a parameter and each column is an individual. We can see that broadly within each of these individuals, there is a similar hierarchy of SHM hotspot motifs. So GYW is at the top, whereas GRS and SYC are at the bottom. They're the coldest. There's also a lot of variation in omega, DNDS, and the CDRs. If we look more closely, we can see that there is significantly lower WA hotspot mutability in older subjects for both male and female subjects. So this is potentially evidence for altered somatic hypermutation machinery with age. Another thing we looked at is signatures of selection during influenza vaccination. For this, we have a data set of 3 subjects sampled at 10 time points over the course of influenza vaccination, and we estimated substitution model parameters for each of those. And one thing that really jumps out at us is this really high increase in mean tree length at day 7 for these subjects. So mean tree length is indicative of longer trees, as in trees with more mutations, and is consistent with a burst of genetic diversity, which is what we would expect from the peak plasmablast response at day 7 after influenza vaccination. When we look at selection, we see something kind of interesting. Generally, we think of affinity-driven selection as positive selection, as trying to find amino acids that increase binding affinity. And the emphasis is on changing amino acids. But what we actually see in these subjects is a significant decrease in DNDS at this time point. And this is indicative of increased negative selection and is consistent with the lineages in these repertoires being memory recall lineages that have possibly already developed binding affinity and benefit less from random amino acid changes. So this is one example of a relationship between mean tree length and lowered DNDS, and we wondered if this was really a more general pattern. What we see when we take all of the repertoire estimates that we performed in these data sets and compare the mean tree length versus the estimate of DNDS, we see that for all of these subjects, there's a very consistent negative relationship between mean tree length, so the length of these phylogenetic trees, and the estimates of DNDS. So this indicates that the repertoires with longer trees tend to have greater signatures of negative selection. And this is in 57 repertoires from 30 different subjects of different age, sex, and vaccine status. And our hypothesis for this is that as affinity maturation continues, antibody evolution becomes more constrained. They benefit less and less from random amino acid changes. So while I'm here, I wanted to plug some of the other analyses we're doing. So a major point of interest is using phylogenetic lineages to explore the relationships between different B-cell subsets and different tissue locations. So given samples from different tissues or from different sorted cell subsets, can we use B-cell lineages to tell us something about how these types relate to each other? And one way that we've developed to study this is given a lineage tree that has been reconstructed that has different cell types labeled at the tips, these could be cell subsets or tissue locations, we can use an algorithm such as maximum parsimony to predict the states at these internal nodes. In addition to being kind of a cool way of visualizing this, it allows us to pinpoint where on trees switches between these types are expected to occur. We can then compare the types of switches that we see in our observed data to the types of switches we see in lineages from permuted tips. One example of an analysis that we've been able to do with this approach is a collaboration that we've done with Susan Marr's group at the NIH, where we've used these lineage trees to try to pick apart the relationships between different memory B-cell subsets and germinal center cells. But this is just one example. Really, this approach is quite expandable. There are a lot of caveats and it's still under development. And if this is something you're interested in, please get in touch with us. And with that, I'd really like to thank Steve, as well as my PhD supervisors, Ollie Pybus and Herten Lunter, as well as our collaborators, Jason Vanderheiden and Julian Zhu. All of these features, at least from the first part of the talk, are released in IGFIML. So if you're interested in doing this kind of stuff in your own data set, you can download that. Do check out my poster if you have any questions. These are my emails, or mine and Steve's emails, and big thanks to our funders.
Perfect timing, Ken. We have time for a question if anybody has one.
I just wonder whether another aspect of changing selection might be an idea that Jose Faro proposed years ago, that in generating memory, you're not just, or perhaps not particularly, focusing on what you've just seen, but rather what you might see. In other words, so there might be a relaxing of the selection process at the end of clonal expansion and the generation of a host of variants that might be valuable in the future. Is that perhaps relevant, or am I off the beam?
I'm not sure what pattern that would generate with this kind of analysis. I wouldn't interpret increase or signatures of negative selection as a decrease in selection generally. So yeah, I'm not entirely, I would have to think about that. Maybe we could talk at my poster.